Welcome to the developmental patterns in animals. In this talk, we're going to learn a little bit about the animal world. In this talk, we're going to start off with learning about the major branches of metazoans, followed by differentiating between Diploblasts and Triploblasts. We'll then learn about Protostomes and Deuterostomes, and thus we're going to learn about different characteristics of the various groups in metazoa.
Now, before we actually look into specific developmental patterns in snails, let's just look at general developmental patterns in animals. Now, all multicellular animals, whether they are vertebrates or invertebrates, come under the group of metazoa. Now, all metazoans, which basically means all animals, undergo gastrulation as part of their development. We normally do not observe gastrulation in plants, for example. There are differences and similarities in the development of different metazoans. And so based on these, we actually have four major branches of metazoans. There's the Basal phyla, the Lophotrochozoan protostomes, the Ecdysozoan protostomes, and the Deuterostomes. So let us look at what each of these branches are.
Let us now look at the first group, which is the Basal phyla. The members of this group have two germ layers and hence are called as diploblasts. When we look at the figure and focus on the one that is to the left, here we can see the arrangement of cells in a diploblast. And the two germ layers, the first one being the ectoderm, can be observed as the cells that are in the shades of blue, which are on the outer part of that organism. The endoderm, on the other hand, are the cells that are shown in shades of gray, and they're more interior in the body of these organisms. Now, members of the basal phyla show radial symmetry, which means they don't have a right or a left part. They're radially symmetric, and hence there are many axes of symmetry that can divide the organism. They also do not have a mesoderm. So if we go back to the figure to the right shows the body plan of a triploblast. And there in shades of red, we can see the cells that form the mesoderm. This germ layer is absent in the diploblasts. Now in the diploblasts, between the ectoderm and the endoderm, we can have a jelly-like substance which is called as the mesoglia. However, please note that these are not made of cells and hence they are not a germ layer. Thus, the only two germ layers present in basal phyla are the ectoderm and the endoderm. The different organisms that are present in the basal phyla belong to the group of Cnidaria, and examples of such organisms are jellyfish and sea anemones, and also organisms that belong to the phyla Ctenophore, and examples of these are comb jellies.
Now, protostomes, which is the next group, are triploblasts that show bilateral symmetry. So, when we say that they're triploblasts, we mean that they have three germ layers. And when we say they have bilateral symmetry, it means they have a right and a left side, like us. In the case of protostomes, the mouth forms first during gastrulation. In fact, that is what protostome means. Proto means first, stoma means mouth. Thus, the mouth forms first during gastrulation as compared to the anus that forms later. Within the protostomes, there are two main groups. The ecdysozoan animals are animals that molt their exoskeletons, and thus animals like arthropods, millipedes, and arachnids come under the ecdysozoan group. The second group is the Lophotrochozoans. Now, all members of this group undergo spiral cleavage, which is a type of cleavage that happens during embryonic development. And they have a common larval form that's called as the trochophore. Now, the example of Lophotrochozoans are annelids, mollusks, flatworms, etc. Let us now look at Deuterostomes, which are also triploblasts that have bilateral symmetry, which means they once again have three germ layers and have the right and left sides, which is similar to protostomes. So what's the difference between deuterostomes and protostomes? It is when the mouth appears. In the case of deuterostomes, the mouth is formed after the anal opening, and thus the anus forms first. Thus, deuterostomes, the mouth says that the mouth forms second. These include the animals that belong to the group of chordates, that's including all vertebrates, as well as echinoderms, which are basically our starfish and sea urchins. Now, to show the differences between protostomes and deuterostomes, let's look at the figure that's shown to the right. If we look at the middle panel, where it says coelom formation, in the case of the protostome, as well as in the deuterostome, we're actually seeing the positioning of the mesoderm, the endoderm, and the ectoderm during embryonic development. Now, in the case of protostomes, we can see where the blastopore is formed, and the blastopore is also formed in a similar region in the deuterostomes. However, when we look at the fate of that blastopore, in the case of the protostomes, that blastopore becomes the mouth, and the anus is formed later during development. However, in the case of deuterostomes, that blastopore forms the anus, and the mouth forms later. Thus, the difference between protostomes and the deuterostomes is whether the blastopore will form the mouth or the anus.
With this, we come to the end of our talk where we learned about the major branches of metazoans, followed by what are diploblasts and triploblasts, as well as how to differentiate between protostomes and deuterostomes.